Okay, we are recording now. It's today the 23rd? Yeah. All right. Abigail. Right in front of me. Yep. Gotcha. Kincaid here. Andrea is online. This really quick. Anyway, Mullins is here. Lewis, yet. Cooper, no Cooper. Hunter is here. Melton is here. Crowder is online. Art, not here yet. Isabel's not here yet. Unless, okay. Julia. Kennedy is here. Hannah. Larissa is here. Madeline here. Hughes, not here yet. Galloway, not here yet. Cook is here. Durgan is online. Spade is here. Burton is online. Kinder, not here yet. Jones, not here. Murray, not here. Hobson is here. Fisher is online. Did I miss anybody? Still have 2 minutes. I won't announcements yet. Wait till everyone. See if I can think of some unimportant announcements. Reminders, it's more like the case. So one of your class on Wednesday, I've been saying it a lot, but I don't mind, because I know a lot of people, they don't really care about independent work until they start on it. They start asking the questions. So whatever, I don't mind. I'm independent work. Just write whatever you want as long as it's about biology, and it can be as long as you want. I really do suggest writing short papers, and I'll have a bit about that here in a second. But for now, I really do recommend nice short papers. I was wondering why this. I looked it up, and according to this source, that's the reason, right? Nice and easy. Accidentally plagiarizing, and if it, write a couple sentences or a sentence. What else? Make sure you do that. You just keep turning stuff in until you hit the 10 points per week, 100 points per semester. Somebody's already hit 100 points. Anything past that is extra credit. Just the route. All right, it's 8. We had some more people come in. Abigail is here. Kincaid is here. Andrea Mullins is here. Is not here. Cooper's not here? No, Cooper's not here. Hunter is here. Caleb is here. Crowder is online. Reinhardt is not here. Isabel is not. Let me back it up here. Lewis is here now. Anyway, Reinhardt's not here. Isabel is not here. Julia is here. Talbert is not here. Kennedy's here. Hannah, not here. Larissa's here. Madeline's Hughes is not here. Galloway is here. Cook is here in red. Durgan is online. Tate is here. Kinder here is not here. Canterbury. All right, so important announcements, I guess. First, with the attendance, a lot of you, I sent out an today email because people have a poor grade, and you want to know specifically what dates you're missing. Else for that matter, reply to the email because you can make dates up. Well, first of all, maybe I made a mistake, right? So you ask me, which dates am I missing? I'll send that back to you. Emails. I sent an email, which I plan to do anyway, eventually, but you can check yourself and let me know if I messed up. One, two is, I probably didn't. You can make up for those absences by watching the videos and follow the instructions. Make sense? Speaking of following the instructions, the first word for red, like the color red, we're watching this online, watch the video and red, take a picture of something red in your room and send that instead. Anyway, so that's the attendance thing. Any questions about it? So remember, you have to be here. You have to get full credit. You have to be on time, and you have to, leave at the end, not early. The other thing, I'm sure some of you have issues with AI. So all of a sudden, my AI detector is flat that says it's written by AI. And I got really suspicious because I'm telling you, every single paper, all of a sudden, it says it's written. So I started testing my own. And then I got it was saying, oh, no, that was written by humans. So now I'm really putting it to the test, and I'm writing papers about biology. Now, every time I write a paper about biology, it flags it as written by AI. Now, I wrote it, so I know it's not written by AI. So the only reason I'm telling you guys this is because you don't have to worry about it. That just means I have more work to do. This is going to take me longer to grade it and I have more. That's why, I'm just letting you know, AI said you didn't write it. I don't believe it because I've tested the AI. So you just know that's why it's taking me longer to grade your papers. Does that make sense? All right. Does anybody have any questions about anything? Jump back into. All right. 1 second. Let me. Yeah. Get. Okay. So let me back up. Finish talking as cellular respiration. And remember, cellular respiration happens in an aerobic process. You have to. Let me back up really quickly as a quick review before we jump into fermentation. So this is what we talked about. This is cellular respiration. We have glycolysis that happens outside the mitochondria. In glycolysis, we break down glucose to make that then, as we know, we already talked about that. That is the mitochondria where it goes into the citric acid cycle, and we continue to break it down, and that's what makes the CO2. In both of these processes, we make a little bit of ATP, but more importantly, we're stripping away these high energy electrons. And then all those high energy electrons come to the big important part, which is chain, oxidative phosphorylation, right? This is where we make the most ATP because we use it from those high energy electrons, ions against their concentration. And again, remember what's happening, and you don't really see it very well in this picture, but all ultimately came from glucose, right? Glucose, which was turned into pyruvate, which was turned into citric acid, so on and so forth. But ultimately, all the glucose, does anybody remember that molecule? I need to know. It's a little the molecule that accepts those electrons in this process. Good. Yes, oxygen, right? So oxygen electrons flow to. And just like any other circuit, any other electrical circuit, if the electrons, like if you cut a wire, the electrons stop flowing. So not oxygen stops this process. Which brings us, let's talk about fermentation. The learning objectives. You will be able to discuss the fundamentals between anaerobic and fermentation. Oh, that's my, anyway, describe the type of fermentation that and the conditions that initiate the fermentation. Whatever, that's what the book says. Let's jump into it. So again, you need oxygen, right? Because without the oxygen, those electrons don't. You're not making ATP, and if you don't make ATP, you die. But cells can work a short period without oxygen. Now, there's some. Think about like a single-celled organism, a bacteria or something like that. It doesn't need as much ATP, but large multicellular organisms like us, we require more. But the process is called fermentation. You need to know what fermentation is. Food energy. Or a more practical way of thinking. The non-oxygen version of cellular respiration. The stuff we just learned about was what happens when you have oxygen and all those electrons can't. What that process does when you take away the oxygen. This side. All right. After functioning anaerobically, so without seconds, your muscle cells generate ATP by a process called fermentation. Know that number. That's just, I'm not going to go in that. But yeah, just so you know, it's about 15 seconds. And of course, that depends on the person and the muscle cells to what you're doing. But yeah, about 15 seconds. So again, once you deprive your cells of doing cellular respiration, because they have to, right? There's no fermentation. Fermentation relies on glycolysis. That is what you need to know for the exam. So of the three stages, cellular respiration, fermentation only uses the first one, which is glycolysis. And that kind of makes sense, sort of. Transport chain, right? Because that's where we use the oxygen. So obviously, electron transport chain. But yeah, try to remember that it only uses glycolysis. Not directly. Indirectly, it does. Because we were stripping away those high energy electrons via that NADH, right? And it goes and it drops off those electrons. And it is, those things, those NADH, they really are like a bucket. You put the electrons in, it takes it to the electron chain, it dumps them out, and then that bucket's empty again. So it can go back, get some more electrons, go back and dump it. You see what I'm saying? It really is. That's what happens in glycolysis. However, it's dumping them out into the electron transport. So without oxygen, where's it going to dump the electrons, right? And that's another thing you do need to know. Well, I've already told you, but glycolysis does. But again, I'm more interested in, it produces a little bit of ATP. I'm not very big on that number is. So any questions about this slide? Again, like I just said here, NAD plus, right? NADH, that's the thing that's carrying the electrons. NAD plus is technically the empty bucket to accept those electrons. To have NAD plus to receive those electrons. And again, that's the respiration. You take the electrons, you go to the electron transport chain, the electrons are flowing, your bucket can come back and electrons. Because that NAD plus is recycled, basically, right? You take it, dump it, it's empty again. It's ready to get oxygen. The normal process. Normally, we strip away these high energy electrons. We dump them at the electron transport chain when they flow down and meet oxygen, water. But in this case, if you get rid of that oxygen, therefore, because we don't have any way to put those electrons. So, recycling the NAD plus cannot occur in aerobic conditions, at least not the way I just described it, right? There's no oxygen to accept the electrons. I find it interesting. I understand why somebody else wouldn't. But here's where we get the process. So since plus, we need those, so to speak, and we have nowhere to put the electron. The pyruvate. So again, picture it. In glycolysis, we have the glucose molecule. We break, and we have two pyruvate molecules, right? That's what you learned when we talked. And now, nowhere to go. That pyruvate. So we've dumped it, right? Now ready to get more electrons again, stores AD plus, and that keeps glycolysis working, right? The other part, this is where it's also a little bit different. When you add electrons to pyruvate, that makes something called lactic acid. It also makes something else, but we're talking about your muscle cells, right? This is fermentation. So you need to know that the product of fermentation in human muscle cells is acid. You need to know that. Obviously, you should know the purpose too. The purpose is to make ATP. The reason that we're doing it this way is because there's no oxygen. You need to know that too. And the product is lactic acid. And maybe I'll go so. And maybe you should remember that it's because we've got these electrons back to the pyruvate. But what I definitely want to ask you, but it's worth mentioning, is this last bullet point here, which is that lactic acid goes to the liver, where the liver converts it back to pyruvate. Sort of continue. So any questions about this slide? All right, the next word for attendance, like BIG. So if you guys want to send them in for extra credit, feel free. For those of you online or watching the video, send a picture of the biggest thing in your room. All right, this is a picture from your old, but it's a good picture that shows what we're talking about. This is fermentation in human muscle cells, right? Again, we've got the 6-carbon glucose. We break it down into pyruvate. We're stripping away high-energy electrons. So far, this seems just like cellular respiration, except these high-energy electrons used to go off to the electron transport chain where they dump it, and we'd have the empty bucket again. But there's no electron transport chain because there's no oxygen. So we have to do something with these electrons. What do? We dump it back to the pyruvate, and we form something called lactic acid. Or lactate. You can call it either one. But I'm going to work with, I think lactic acid is the word I'll use in the exam. So any questions about this slide? All right. Moving forward. Oh, if you download the PowerPoint, watch this little animation. It's I think it's a good little animation to show you how it works. So like I mentioned earlier, we humans and a lot of other organisms, we need the full cellular respiration, right? The full aerobic cellular respiration where electron transport chains making all that ATP. We need that. We can't live off of glycolysis alone. Like I said earlier, sometimes these smaller organisms, like microorganisms, they can. And I'm giving you this as examples. If you're taking notes, give your hand a break. You do not need to know any of this. It's just interesting to know. Lactic acid from yeast can make things like cheese, sour cream, yogurt, soy sauce, pickles, cabbage, olives, sausage meat products. You can look that up if you want for independent work. Maybe you can find some other food products that are made from lactic acid. Or you could look up actually how they do. Okay, you know that lactic acid is used to make cheese now. But you could look up exactly how is lactic acid used to make cheese. But again, that won't be on the exam. Yeast. This one's a little bit more important for the exam anyway. Does anybody know what yeast is used for in this context? When you think of yeast, what do you think of? Actually, I shouldn't say in this context. If you weren't in a biology class and you were just, I don't know, talking to your parents or somebody, they were talking about using some yeast. What are they using it for? What would you use yeast for?
To make bread.
Online and in person said it. Yeah. Bread is one thing. That's a big one. What else? No one? Alcohol. Anyway, yeast. This is a very interesting organism. It is capable of regular old cellular respiration, aerobic cellular respiration. So cellular respiration with oxygen. If yeast has oxygen, that's what it does. Because, and I don't remember if your book gets into this or not, and I'm not going to ask you, but the interesting thing to me about yeast is it is a single cellular organism, but it's not a bacteria. So it is a eukaryotic organism. It does have that. It has organelles, right? It's larger. It's more complex. So it does need cellular respiration a little bit more than a bacteria would. It's still single-celled, but it's more complex. And that's what, in my opinion, what makes yeast so interesting. So if you give it oxygen, it just does regular old cellular respiration. If you cut the oxygen off, then it does fermentation instead. And that's how we make alcohol, right? That's how we do it. We basically starve the yeast of oxygen, sugar in the corn mesh or the potato juice or whatever it is that you're using to make your alcohol. It breaks it down, all that sugar in there, and it converts it to alcohol. But for the exam, all that information's on. For the exam, what you need to know is this. So remember, the first type of cellular respiration we talked about was cellular respiration of muscle cells. All right, 1 second. Sorry, guys. All right, anyway, cellular respiration of muscle cells produce lactic acid. Remember, we're talking about the exam here. The other type of cellular respiration, you need to know this for the exam, produces alcohol. So there you go. For the exam, all that talking, that's basically mostly what you need to know. Cellular respiration in muscle cells produces lactic acid. Cellular respiration in yeast produces alcohol. And I'm not going to try to get tricky. So there's a few ways I could write that. I might write alcohol. I might write ethyl alcohol. I might write ethanol, whatever. I'm not going to try to trick you, though. There's not in this multiple choice exam, there won't be like different types of alcohol listed. We listed alcohol listed once in the multiple choices, and that will be the correct choice. So any questions about that? And once again, if you're into this thing, you could look that up for independent work. I just told you the yeast is used to make alcohol, but I didn't tell you how. So you could look that up, I guess, if you want. Any questions about this slide? Here's a picture of what I'm talking about. Again, this is from your old textbook, not this new textbook. But once again, we're using glycolysis, right? There's only a slight difference. Here, once again, we start with the 6-carbon glucose. We break it down into 2 pyruvates. Once again, we're stripping away high-energy electrons. Once again, those high-energy electrons have to go somewhere. So where do they go? They go back to the pyruvate. And in this case, before that happens, or after, whichever, we also release some carbon dioxide, right? And then we're left with ethyl alcohol. So that's another difference. I don't know if I'll ask you or not on the exam, but another difference between the first fermentation we talked about and this type. The type that produces alcohol produces carbon dioxide. An easy way to remember that, I don't know how easy this is, but think about something like beer. Beer's been around for a long time. Actually, this is a good end to look it up. Beer saved thousands of lives when it was first invented. Water was garbage in that part of Europe. And they were just making beer to get drunk, but the process of making beer also kills all the microorganisms, so it was good for them. Anyway, what was I getting at? Beer, oh yeah, a way to remember this produces carbon dioxide. So remember, beer is carbonated. And if you're of legal age, we could go study that together. But no, beer is carbonated, and that's where it comes from, right? It comes from that. Another way to remember this is like somebody, I think Michael said it. We use yeast to make bread, right? And bread rises. You guys know that, right? Well, this is why it rises. Because the yeast, when it's doing this, is producing dioxide. I hope so. Anyway, let's talk about this really quickly. Book included it, so did I, but I'm not going to test you on this. But some prokaryotes, for example, certain bacteria in our case, use anaerobic respiration. Actually, I wonder if I wrote that down wrong. I misread your book. That's almost all prokaryotes do this, but anyway. Oh yeah. So then there's some that do something a little bit different, right? The methanogens, like the archaea, carbon dioxide and methane while oxidizing in ADH. What I'm saying, when I say oxidize in ADH, when you oxidize something, that's getting rid of the electrons. It produces methane instead of lactic acid or ethanol. Reduce sulfate to hydrogen sulfide, regenerating the NADH. That's from your textbook, so I included it, but I'm not going to ask you any of that. There's two types of fermentation. One of them produces, the other one produces alcohol. Any questions? Let's see. The third word already? I think it's the third word anyway. The next word for attendance is small. So, if you want the extra credits at the end of the class, if you are online, this thing in your room. All right, we'll move forward. So again, this is from your textbook. This is what, but I'm not going to test you on this. I really want to focus on more of a human aspect. But anyway, fermentation of bacteria. Some require oxygen, right? Some bacteria absolutely have to have oxygen. Some that can switch back and forth, sort of like yeast, right? Yeah, and that's basically what we're saying. And again, I'm not going to ask you that. So I think about what I said about yeast, right? Yeast perform regular cellular respiration. They can also perform, and some bacteria are the same. And again, I'll put a big old X to this visual reminder that will not be on the exam. All right, some fermentation characteristics. So putting the words in a form. All fermentation uses gas, and I'm only going to test you on the two types of fermentation, right? So there's the one that produces lactic acid, and then there's the other one, which I already told you is the one that produces alcohol and carbon dioxide. But your book does point out the gas production is used in lab bacterial identification. I'm not going to ask you that, but that is one of the ways, right? So in labs, there's different ways to figure out what kind of bacteria you have. That's one of them. You can one of the ways is by identifying their gas. Again, the importance of fermentation. Just a reminder, this is important, right? We need that NAD+. We need that empty electron bucket, right? For glycolysis to happen, we need that thing to strip away the high energy electrons, which means we need a place for those electrons to go. And since we don't have oxygen, they go back to the pyruvate, right? Because without that, we would not have ATP and we would die. Any questions about this slide? Okay, here we are. This is good. This is the last. We might have an early day. It was great for a Friday. Anyway, the last bullet point for this chapter is connections to other metabolic pathways. And to be honest, this is probably one of the least important parts of the chapter, so that's good. Anyway, by the end of the section, here's the learning objectives. You should be able to discuss the way in which carbohydrate metabolic pathways, glycolysis, and the citric acid cycle interrelate with protein and lipid metabolic pathways. That's a really fancy way of saying, you know how we keep saying we use glucose in cellular respiration? We break down glucose to make ATP. This bullet point saying, well, let's talk about the other stuff. Like how many of you ate today? Or how many of you have eaten in the past 24 hours? How about that? Okay. Did any of you, when you ate, did you have like a container of pure glucose? Did any of you eat pure glucose? Nobody, right? You probably had some proteins, maybe some carbs, right? Maybe some fats. Point is, here we are, I keep talking about glucose, but think about it. We eat so much other stuff. We eat other stuff. So we're going to talk about how that works then, right? Anyway, and then we'll also be able to explain why the metabolic pathways are not considered closed systems. So here we go. Respiration is versatile, right? So I keep saying, and for the exam, you should just focus on glucose. For the exam, in your mind, you should think cellular respiration breaks down glucose, right? In your brain for the exam. This off to the side, just as a side note, let's be realistic here. And again, so we're going to talk about how those things are put to use. But here we go. This is a picture from your old textbook, but it's a good one. So again, we eat food, right? We don't just eat pure glucose. What kind of food do we eat? Well, usually it's carbs, it's fats, it's proteins. Those carbs, that one's the easy one, right? So as you should know by now, for example, starch is a bunch of glucose molecules, right? It's a bunch of glucose molecules put together. So that makes sense. In a sense, carbs are glucose, right? They're just attached. So you break them down, and like we said, we have glucose, right? Glucose goes into glycolysis. Fats, on the other hand, as you know from the chapter 2, up of a glycerol molecule and 3 fatty acids, right? So when you digest it, those get broken apart. The glycerol itself goes into glycolysis, and the fatty acids, depending on what kind of fatty acids, the fatty acids go into this little middle step between glycolysis and citric acid cycle. Proteins, remember, proteins are made up of amino acids, 20 different amino acids, and they're all very different, right? So depending on the protein, depending on the amino acid, it's going to get broken down, and it could go into glycolysis. It could go into this little middle stage between glycolysis and citric acid cycle, or it could go directly to the citric acid cycle. So some test questions on here could be like, which stage uses carbohydrates? And the answer would be glycolysis. Which stage uses fats? And I guess you could say glycolysis and the stage between glycolysis and citric acid cycle. Which stage uses proteins? And then the answer for that would be all 3. Another question could be which is the only stage, or excuse me, how am I going to word this? Which stage only uses proteins? Well, that's not necessarily true either. Depends on how you look at it. But if you see here, proteins are the only ones that can go directly into the citric acid cycle. They're go through this process first. But anyway, most likely I'm not going to ask you any of that. But if I did, those would be the questions I asked you, right? I would ask you, where are sugars used? Where are fats used? Anyway, any questions about that? Okay. So the last word for attendance, since the screen is black, will be black. So for you guys, you can send me the word black for extra credit. If you're online or what, send me a picture of something that's black in your room. And that's it. Early day on a Friday. We'll see you on Monday when we start learning about photosynthesis. I'm going to try to get a lot of work done today, so keep an eye on your emails. Oh, and if you came in late, make sure you email me.